Today I will talk about the structure and function of alpha-1 antitrypsin, an inhibitor, a proteinase inhibitor circulating in the blood. Site and synthesis of alpha-1 antitrypsin, the isoforms that are known to be produced by mutations of the gene, and the consequences of antitrypsin deficiency, liver, kidney, and lung disease. Alpha-1 antitrypsin is a serum protein. What does it mean? It means that it is synthesized and released to the serum, and it functions there. Serum proteins in general are of two kinds. One is, like alpha-1 antitrypsin, has a function in the serum or in the blood. All of these serum proteins, except for the immunoglobulins, are synthesized by the liver. Please remember that for the future. There are additional proteins circulating in the blood that are not functioning there, but they are released by other cells, most likely by cell death. You may recall that there are certain enzymes, certain proteins that can be used for diagnostic purposes by detecting the elevated level of these proteins in the blood. For example, you learned about myocardial infarction. Some of the enzymes are released by the heart cells, including creatine kinase. A certain isoform of this particular enzyme is produced only in the cardiomyocytes, so the increased level of this enzyme in the blood is diagnostic for myocardial infarction or the death of the myocardial-cytes. We have 7 to 7.5 grams of protein in the blood. So the blood is pretty rich in protein. These proteins can be separated from each other based on their size and their chart. We have a huge peak of the electrophoretic in the electrophoretic pattern made only by albumin. Albumin is about 50% of our serum proteins, and albumin has a lot of functions that you may have learned about until now. It ensures the proper oncotic pressure of the blood, and it can bind also hydrophobic molecules and transport those molecules in the blood. Other peaks in the electrophoretic pattern are labeled alpha-1, alpha-2, beta, and gamma. And under these peaks that we can quantify actually, run a lot of different serum proteins. Alpha-1 antitrypsin was named by the fact that 90% of the proteins that run under the alpha-1 peak is actually this serum molecule, alpha-1 antitrypsin. The function of the alpha-1 antitrypsin is being a proteinase inhibitor. It inhibits proteinases, enzymes, that are released especially by white blood cells to the serum. The strongest inhibition by alpha-1 antitrypsin is against elastase, but it also can inhibit different collagenases, trypsin, and chymotrypsin. How does this inhibitor inhibit enzymatic activity? In the reactive site of alpha-1 antitrypsin, we have a methionine. This methionine that can bind irreversibly to different proteinases. It acts as a suicide substrate. What does it mean? It means that it binds to the active center of the enzyme, and the enzyme actually can cleave the inhibitor. However, when the inhibitor is cleaved, it binds even more strongly to the enzyme. So the enzyme will not be active anymore, and it will be removed by the reticuloendothelial system from the serum. Thus, alpha-1 antitrypsin protects the tissues from being attacked by these proteinases. So what is wrong if this inhibitor is missing from the serum? Well, definitely the inhibitory effect will be missed, which is a big enough problem itself. However, alpha-1 antitrypsin deficiency is affecting multi-systems, multiple organs. Why? Well, some of the mutants of alpha-1 antitrypsin is actually such that it inhibits the secretion of the protein from the liver, and this buildup of abnormal protein will damage the liver cells. It can also result in, of course, absence of protein function, less circulating in the serum. And then it can be a reaction of the body to the accumulated abnormal proteins being removed completely from the liver by intracellular actions, or it can cause problems in other organs. So let's see the data. The alpha-1 antitrypsin, as every protein, is known in several different isoforms. Most of these mutations are benign, which means that yes, there is a change in amino acid, but it does not result any change in function. M is the normal or wild-type function of this alpha-1 antitrypsin inhibitor, and this is the form which is working properly. However, some mutations alter the function of the protein. One of these mutants are called Z. Although you don't need to remember, but I wanted to show you that in this case, a glutamic acid which is charged negatively changed to a lysine which is positively charged. As in every protein, if the charge is changing, there may be different interactions within the protein or among the secreted proteins, and this is what happens in the Z mutation. The newly synthesized alpha-1 antitrypsin protein actually polymerizes within the cell, and it impairs the function of the liver cell, but also impairs the secretion of antitrypsin in the liver. The S variant, which is a glutamic acid changed to an apolar valine, actually is recognized by the machinery within the cell as an abnormal protein, and it is removed intracellularly. Some of these proteins, however, the Z variant and the S variant, is secreted out to the blood. The PI mutation is this methionine in the active center of the inhibitor is changed to an arginine. It's a very peculiar mutation because this mutation renders the inhibitory function zero. It's not functioning anymore, but this arginine in the same point actually converts the inhibitor to a thrombin inhibitor, which means that this inhibitor will inhibit factor 2 or thrombin, the most important blood coagulation factor, and thus it may cause bleeding disorders, on the top of not inhibiting the other proteinases, which the antitrypsin should. And finally, Q is the null allele, which means that there is a big deletion in the gene. There is a truncated protein that is synthesized, and actually this protein cannot be synthesized because of the deletion, or it right away eliminated by the system. Two of the most common mutations are Z and S. Consequences are liver disease, kidney disease, lung disease. So let's see how it is developed. The Z mutation is the one where there is impaired secretion from the liver because of polymerization. So large amounts of this polymerized antitrypsin accumulate in the liver. when there is a homozygous individual with ZZ mutation, liver is significantly damaged. Interestingly, the kidney is damaged, perhaps because what is secreted out, a small amount, from the liver, it then accumulates in the kidney and damages the kidney. So some of these Z mutation proteins are released from the liver, but it only gives 10 to 20% of the normal circulating protein level. And also this mutation somehow compromises the inhibitory function of the antitrypsin. This leads to lung disease. The S variant, which goes through rapid intracellular degradation, again, some protein is secreted, but it is in a very much decreased serum level that we can measure. It is working normally, but we have less, so the result is most of the time is lung. Why lung disease? What you see in an alpha-1 antitrypsin patient is pneumothorax, asthma-like symptoms like wheezing, problem with pushing out air, hyperexpansion of the chest, shortness of breath, and chronic cough. All forms of alpha-1 antitrypsin deficiency can cause these symptoms. For some reason, not all patients are getting these symptoms, but we don't know what the reason for some patients not to develop lung disease. The onset in age is variable, typically in the teenage years or later, and it has a slow progression in the case of ZZ or SS carriers. The null mutation, of course, there's an earlier onset because there is actually no inhibitor circulating in the blood. So what is the reason for this? Of course, lack of function, lack of the inhibitor or decreased amount of inhibitor which circulates in the blood. Under normal conditions, lung macrophages release lots of proteinases. This is a defense mechanism towards intruders that we inhale with air. It can be dirt, it can be toxins, it can be different microbes. So it's normal that macrophages release proteinases. However, we need to have enough inhibitors secreted and being in the lung to actually have a good balance between the proteinases and inhibitors. Now, lack of inhibitors, lack of antitrypsin, because less is secreted by the liver, causes an imbalance, and these excess proteinases chew up the lung tissue, especially elastin, which is giving the elastic property for the lung by elastase, which is released by macrophages. If elastin is lost, the air is trapped more in the lung. It is difficult to push air out. Several times you see the alpha-1 antitrypsin patients to purse their lips and force the air out of their lung. Very interestingly, alpha-1 antitrypsin deficiency, this destroys mainly the lower lobes of the lung, while in normal emphysema caused by mainly smoking uniformly destroys the lung. So what is the relationship between smoking and lung disease? It's twofold. Smoking irritates the lung and brings in more pollution, and more macrophages will go there and to try to destroy all. So more enzymes will be released by macrophages. Smoking, however, also affects negatively the macrophages. Smoking stimulates neutrophils, macrophages. They also release, especially the neutrophils release different oxidants. You remember oxidative burst. which will destroy the methionine residue in the center of the inhibitor. So it is not enough that more proteinases will be released in the lung, but there will be even lower number of inhibitors because of this destruction by smoking available to inhibit the proteinases. Emphysema develops in many homozygotes, ZZ, SS, or Q mutants. How do we diagnose? This is for your information only. Of course, clinical features are very typical of alpha-1 antitrypsin, but we can detect aberrant proteins, or we can phenotype the proteins and we can phenotype the gene to see where the mutation is. Liver biopsy is taken, and it is especially typical for ZC mutants to see the destruction of the liver and accumulation of this protein in the. How can you treat the patients? Again, it is for your information only. Protein replacement therapy is known, either normal purified alpha-1 antitrypsin from normal patients, or recombinant protein is also available to be given to patients with alpha-1 antitrypsin to inhale and save the lung. In ZZ mutations, liver transplant is sometimes necessary, sometimes even kidney transplant. There are efforts to transfer the gene into the liver of the inhibitor, and stem cell treatment is also being developed. So that is just for fun epidemiology of alpha-1 antitrypsin deficiency. Actually, it comes from Europe. The Z allele is very frequent in Northern Europe. You see it is Estonia and Latvia and Denmark, where the highest number of Z allele carrying patients are there, but it is also frequent in the rest of Scandinavia. The S allele is coming from the Iberian Peninsula, and then it spread to Europe. For some reason, the frequency is a little bit higher in the United Kingdom. So in summary, you learned about an inhibitor which naturally circulates in our blood and saves our tissues from being chewed up by different proteinases. This is an inhibitor within a methionine in the center, which can bind covalently to the target proteinases. This is, as every serum protein except for the immunoglobulins, synthesized and released in liver. M is the normal form. The most frequent mutant is Z, which is polymerization of the aberrant protein in the liver, affecting the liver, affecting the kidney, and because less is secreted, affecting the lung. S is a mutation result in a protein which is removed intracellularly, so again much less is secreted to the blood. And the Q is the null mutation, which again less or none is circulating in the blood. So liver and kidney disease develops in Z and sometimes SS mutants. Lung disease can develop with all mutants, but not every patient will actually develop lung disease. The reason for lung disease is lack of inhibition. Much less or no inhibitor is circulating, and the proteinases, which is normally secreted by macrophages, are not balanced by inhibitors. Smoking contributes to lung disease because smoking results in more release of proteinases by macrophages, and also destroys the inhibitor by destroying this methionine residue, covalently binding methionine residue, not being able to inhibit the inhibitor, the antitrypsin not being to inhibit the proteinases. Thank you for your attention.